Good afternoon, students of St. John's College of Pharmaceutical Science and Research, Kattapana, Idiki, Kerala, a 5th semester student. I would like to continue the Unit 1, in which some of the secondary metabolite biosynthesis have to be included. Already in the last two classes we have seen most of the primary and secondary metabolites and secondary metabolite pathways, basic pathways like shikimic acid pathway, mevalonic acid pathway, acetate pathways, and various steps in them and various enzymes which is responsible. And we have discussed also what all the products which is available, which can be produced by these pathways. And also we discussed about sometimes some products are going to 2-3 channels. They may pass through shikimic acid pathway and also acetate pathway also they will pass. Today actually it's a short discussion, but it is very important how these pathways are responsible for the production of secondary metabolites like glycosides and flavonoids and some aliphatic amino acids also. So here in this chapter we can have the discussion about the biosynthetic pathway of glycosides and amino acid. I did not mention the flavonoid because flavonoids, most of them, they have some glycoside linkages, and so that it will included in that, and we will have a discussion about that. So now you see here biosynthesis of glycosides. As I told in the beginning, biosynthesis of glycosides, the metabolic process of glycoside formation occurs in two steps. In first step, various types of aglycone are formed by biosynthetic reaction, whereas in second step, coupling of aglycone with sugar moiety occurs. So in this junction, students should know their basic knowledge about the glycoside. Glycoside will have always two parts, a aglycone part and a glycone part. Aglycone part means which has maybe aromatic ring structure, maybe a complex ring structure, alcoholic or aldehydic or maybe phenolic. These type of structures will be there, and which will be linked with an sugar molecule, that is what glycone part. So a glycone part and a glycone part will be there, which will be linked by a bond, usually by oxygen, carbon, sulfur, nitrogen will be there. So this linkage, by linkage of the bond between this A glycone and glycone also, they can be classified as O glycosides, C glycoside, N glycosides and S glycosides. So this is what we have to remember. So once you remember this, even in the higher studies, in your next chapter or something, when you are studying about various type of glycosides, the details about the crude drugs and their constituents, it will be easy for you to remember their structure and its applications. So in different types of glycosides, interaction of nucleotide glycoside occurs between UDP, that means uridine diphosphate glucose, with alcoholic or phenolic group of secondary compound A glycone called O glycosides through linkage with carbon C glycoside, nitrogen N glycoside and sulfur S glycosides. And this is a slide just for your understanding to know that glycosides are having two parts. One is a glycon and a glycan part. You see glycan part here is a glucose. A name is also glucovanillin. It's a beta isomer and alpha isomer, beta isomer. This glycoside, there are linkage of alpha, beta is also there according to their configuration. It can be denoted with alpha and beta. According to the glucose, you may be knowing glucose is having 4 chiral carbon atom. Chiral carbon atom. So chiral carbon atom is there means you will be finding out how many isomers will be there. You will be that is 2 to the power of n will be the formula. You might have studied in your organic chemistry. So it is a beta isomer and also it is a glucovanillin. So you can see that when it is hydrolyzed, they will break down, the glucose molecule will be separated, and the A glycan part, you can see that it is an vanillin which has one OH group and an aldehyde group and an methoxy group is attached in a benzene. So vanillin is the one of the A glycan which is present in the glucovanillin structure. So this is one simple example about the glycoside for your understanding. And here you can see that UDP, that means uridine diphosphate glucose with alcoholic or phenolic group of secondary compound or a secondary metabolite compound A glycan called as O glycosides. So you see the structure here which is shown there, which is present in the seedlings of soya bean, can be converted by UDP glucose to UDP. That means glucose molecule is coupled with this A glycan molecule and you are getting, and we have shown that R in the structure, that is radical. It can be given an option. R can be hydrogen means diarsine, and if it is OH means genistein, and then you can see that is what it is repeated the name in the second time. And also you can see isoflavone 7O glucosyl transferase from the root of soya bean seedling, that is glycine maxis seedlings. This is present in the soya bean seedling and this reaction takes place. That is what we want to know it. In this junction, students should know that glycoside will have two parts, a glycan part and a glycan part. In a biosynthetic pathway, the glycan part and A glycan parts are joined together by means of transferase enzyme. And it can be, it's not necessary it should be only glucose, it can be any carbohydrate. It is also possible to have the linkage with an A glycan molecule to give a glucose, not glucose, sorry, to glycoside. The following two steps are involved, that is which we discussed. That is one, in first step the uridine triphosphate transferred and that is uridyl group to sugar 1-phosphate and forms uridine diphosphate sugar and inorganic pyrophosphate. The enzyme which catalyzes this reaction is uridyl transferase, which we discussed before. So uridyl transferase, it's just coupling. It's just joining the uridine triphosphate. It converted to uridine diphosphate sugar, and the second step is coupled with the A glycan molecule. The second step, transfer of glucose moiety from uridine diphosphate to a suitable acceptor. Suitable A glycan occurs. This reaction is mediated by enzyme glycosyl transferase and forms glycoside. So you say UDP sugar plus acceptor A glycan will gives in the presence of glycosyl transferase, you are getting acceptor, that means a glycon joined with the sugar, and the UDP, that means uridine diphosphate, is removed. And now we are coming to this slide to discuss about the other aglyconic part. Aglycone also formed as a secondary metabolite in the plant. That is also biosynthetic pathway. So the sugar of glycosides or monosaccharides may be ramenose, glucose, fructose, or deoxy sugars, that is digitoxose, or cymarose can also be the sugar part. And the aglycone moiety of cardiac glycosides are steroidal in nature. So there I am stopping, because students should know what is the steroid structure. Basic organic chemistry you might have studied in steroid structure. You see a picture which I have shown here in the right hand side, A, B, C, D, which is denoted with numbering also of that full molecule, which contain 19 carbon atom, which is present there, with including the 2 annular methyl group. So you see there are 3 6-membered ring structure which are named as A, B, C. When 3 6-membered ring structure are there, then it is called as, what is the name of it? 1 means benzene, 2 means naphthalene, and 3 means what you will see, phenanthrene. You might have studied in your organic chemistry, 100% sure. So benzene, naphthalene and phenanthrene. So now we are talking about phenanthrene. This is a phenanthrene derivative, and what is D? D is attached to the phenanthrene, which is a 5-membered ring structure. So it is a penta. It is a pentacyclo ring structure. So what is the name you see there? These are derivatives of, you can see the slide I have written there. These are derivatives of cyclopentano phenanthrene ring structure. So cyclopentano phenanthrene ring structure means A, B, C, D. It is the basic ring for steroidal compound. Understand? And it contains an unsaturated lactone ring attached in C17. See the position of the 17th number, and you see that these two structure which I have given down. One is cardinolite and another one is bufenolite. They are also an aglycone moiety of a cardiac glycosides. So what is the difference between this cardinolite and bufenolite structure? A cardinolite structure, you have 5-membered lactone ring, and in bufenolite ring structure, in the structure of the compound have a 6-membered lactone ring in the 17th position. You can see that 17th position. For just to make it, you should understand the 17th position. I have drawn this basic structure A, B, C, D of an steroidal skeleton. So steroidal skeleton will have A, B, C, D, and the numbering you should remember. This numbering will not change. So this will give you more idea to remember the structure. So now we will come to the slide what is written there. These are derivatives of cyclopentano phenanthrene ring structure, which contain unsaturated lactone ring attached to C17 and a 14 alpha hydroxy group. We can see there is an hydroxy group, 14 alpha hydroxy group in the cis juncture of the C and D structure. So C and D structure, 14th position is there. There is an OH for this bufenolite and also cardinolite. So this is one type of very important for anyone to remember, that is a cardiac glycosides, which will be having a steroidal ring structure of aglycone, which has 17th position. They will be having lactone ring, and in the 14th position they will be having OH group in the juncture of C and D structure. So, and also it is very important for any student should know that, and the steroidal ring structure means it is cyclopentano phenanthrene ring structure. That means phenanthrene is have 6-membered ring structure, which is attached with, or which is just attached in the 14th and 13th position. You can have an 5-membered ring structure, that is called and cyclopentano ring structure which is just present. That is what you have to remember, so that student will not find any difficulty in writing the structure. And now we are coming to the next anthraquin glycosides, which are also biosynthesized by shikimic acid pathway in Rubiaceae family. The alizarin is the biosynthesis, is the product which is shown here. Alizarin is an anthraquin glycosides, will have quinone derivative with OH groups. So you can see that it is an aliphatic. It is not aliphatic, it is an synthesized. It is synthesized by shikimic acid pathway in Rubiaceae family. Alizarin is biosynthesized, shows a ring structure A and derived from shikimic acid. You can see that A, C, two ring structure I have named that. And A is made from shikimic acid and C is made from mevalonic acid structure. So it is very clear there are two pathways are involved to production of this anthraquin glycoside. Example, alizarin. There are many anthraquin glycosides, but we have taken only one example of alizarin, which is present in the Rubiaceae family. So this you can find out how it is present. You can see the mevalonic acid is responsible for the ring structure C, and A ring is formed from by the shikimic acid pathway. Aromaticity will be given. And it is important to understand the biosynthesis of flavonoids due to their diversity, because flavonoids are diversity. It is also having glycosidal linkages which has a multiple structure. These flavonoidal molecules are biosynthesized by their precursor, which is three molecules of acetic acid and phenyl propane moiety. It mainly involves the interaction of 5 different pathways which are named as 1. The glycolytic pathway. Number 2. Pentose phosphate pathway. That means pentose phosphate pathway means, don't think that is a new one. You should know that we have discussed last class itself about the isoprenoid unit. Pentose, isopentyl pyrophosphate, dimethyl allyl pyrophosphate, and about all the cycles about geranyl pyrophosphate, farnesyl pyrophosphate, squalene, how it is responsible for the production of cholesterol. These all things you should remember. And the shikimic acid pathway, the synthesis of phenylalanine, that also we have discussed about how pyruvate and erythrose 4-phosphate and 7-heptilo compound, and then it is converted into quinic acid, and quinic acid is converted into dehydroshikimate, and shikimate is converted into then shikimate. Shikimate is converted into corosmate. Corosmate in this is also can give rise to even prefimate. So these all things which is responsible for production of aromatic amino acid like phenylalanine. And you can see phenyl propanoid metabolism. A phenyl group will be formed by the shikimic acid, and a propanoid group also metabolism which produces active cinnamic acid. Cinnamic acid is derivatives which is 4-coumaranyl CoA and lignin. Also a plant structural compound can be obtained. And fifth one, the diverse specific flavonoidal pathway. So you see, in a flavonoid pathway we should have many structures, a complex structure. We need these pathways, you have to remember. So the questions can be asked by any teacher in different ways. So you should not think that it can be written only mevalonic acid pathway, shikimic acid pathway, acetate pathway only we studied. Almost all of them are depending on them, but still by means of their changes and their precursor changes, they used to name according to the product which is obtained. So there are many pathways, even the question may be asked by different names. So you should have the capacity to understand, and you have to write yourself the pathways and structures. And you see, the flavonoids are biosynthesized via condensation of the shikimic acid, and there is acylpolymalonate pathway. the acylpolymalonate pathway. They did not say directly malonate pathway, they say acyl group polymalonate pathway. And the phenyl propane, that is cinnamic, phenyl propane, we have discussed propane, the cinnamic acid derivative, and synthesized from shikimic acid, which acts as a precursor in a polyketide synthesis. In this scheme, additional 3 acetate residue are incorporated. So, we are going to tell about this synthesis. So, there is 3 acetate groups are incorporated in this structure and followed by ring closure. The plant biosynthesis different classes of flavonoids like flavonols, flavonones and flavones, flavonols or catechins, isoflavones, isoflavones, dihydroflavonols, anthocyanidins and chalcones. Through subsequent hydrolyzation and reduction, you can obtain flavonoids. So, see here, the same thing we have written, what you have explained there, we have shown in a strict structural form 3 molecules of acetic acid, acetate molecule, it's just written in acetic acid molecule, and gives and plus a phenyl propane, and you will get an A polyketide form, and then it gives a chalcone moiety which can be converted into flavonoid, This is just it's not a complete structure. You can see the C bond, it is not a complete structure, it's just a flavonoid. We did not give any name, just to know about the basic structure. It can have many substituent in the carbon atom, and many ring structure can also be substituted in the flavonoidal structure. So, in biosynthesis of cyanogenic or cyanophoric glycosides, example prunacin, the amino acid phenylalanine acts as a precursor. In this biosynthetic pathway, an aldoxim, a nitrile and a cyanohydrin are involved as intermediate. See, I have written aldoxim can be converted into hydroxynitrile by means of enzyme cytochrome P450. And uridine diphosphate glucosyl transferase can convert this hydroxynitrile to cyanogenic glycoside. Very simple to remember. If they ask something about glycoside, you can just write about this. And now this is a repetition slide. Already we have discussed about cardiac glycoside, and the same thing again I am coming to here. The aglycone moiety of cardiac glycoside is basically steroid in nature, we have discussed, and these are derivatives of cyclopentanophenoanthrin ring, which contain unsaturated lactone ring. This ring attached with C17 and 14 beta hydroxyl group and a cis position of the ring C and D. That is why we have written the basic structure also in between C and D. The steroidal biosynthesis is derived from cholesterol production pathway, that is acetate pathway. The biosynthesis of cholesterol involves cyclization of squalene. We have already studied about the cyclization of squalene, an aliphatic terpene. Squalene is an aliphatic terpene. Do you remember how the squalene is formed? Acetate will join with it, it will be another acetate molecule, it will be acetoacetyl-CoA, and another acetyl-CoA will join and it will give HMG-CoA. HMG-CoA is converted into mevalonate. Mevalonate is converted into, what is the main thing you have to remember? We are getting that 5-membered carbon atom compound isoprenoid, that is isopentyl pyrophosphate, and then it can be converted into isomerase dimethyl allyl pyrophosphate. These two 5-membered carbon atom compound will join together and you will get a 10-membered carbon atom compound which is called geranyl pyrophosphate, and another one molecule of isoprenoid unit or isoprene unit when you are joining, that is dimethyl allyl pyrophosphate, you will get a 15-member carbon atom compound that is called farnesyl pyrophosphate. And while discussing about farnesyl pyrophosphate, we talked about head and tail. And two molecules of farnesyl pyrophosphate, they join in the tail to tail joining. Usually tail will be joined with the head, but here tail joining, and you are getting the structure of 30 carbon atom compound that is called squalene, and that squalene will go for cyclization, and then you are getting that lanosterol and then dehydrolanosterol, then comes you are getting cholesterol, and from cholesterol you can find various hormones, progesterone, prednisolone, estrogen, corticosteroids, these all we discussed in the last class. I just made some time to you to remember what we studied before, that's all. And now we see here also the same thing. Even when it is to come to the cardiac glycosides, it has a steroidal ring structure, and it has a basic ring structure equal to the cholesterol. So that is why they have written that name, biosynthesis of cholesterol involves in cyclization of squalene and aliphatic triterpene it is. And then you can see the sapogenin occurs. Now sapogenin glycosides. Sapogenin occurs, it is also a long structure, complicated structure, but it is very simple to remember. It is also a glycoside, so it will have surely a sugar part and as well as a glycan part. A glycan part and as well as a glycan part. So a glycoside forms in saponins, that is the neutral saponins are steroidal derivative possessing spiroketal side chain, and acid saponins have triterpenoidal structure. The pathway is similar for the biosynthesis of sapogenins. The triterpenoid hydrocarbon squalene is formed after cyclization of triterpenoids in one direction and spiroketal steroids in other direction. The squalene, cholesterol and other steroidal compounds include aglycone are formed by the following manner. You can see here a structure, just going down, before going down I have given some structure to know about what is the saponin. Saponin is a glycan, and also they will be having an glycan part. So a glycan part, you see it is a multi-sugar molecule. 3 sugar molecules, 3 glucose molecules are there. Multi-sugar molecules will be present in the saponin. And this is also to say about saponin. Saponin is always studied by it gives some frothing, it gives some soap formation, it acts as a surface acting agent. You might have studied in your pharmaceutic surface acting agent, which reduces the surface tension between two immiscible liquids. That is why you are using this surface acting agent in preparation of emulsion. And that is why you can even why you are using, because one phase should be dispersed in other. So if you want to dispersion phase in a continuous phase, the surface tension between the two immiscible liquids should be reduced. It can be done by surface acting agent. So that type of surface acting property, the physicochemical property, is present in saponin. How? The saponin is having two molecules. One is fat soluble triterpene. You can see that fat soluble triterpene, a steroidal ring structure, and another one is water soluble carbohydrate chain. So saponin, usually you can see hydrophilic tail and hydrophobic tail and also hydrophilic head will be having a typical surfactant molecule. So you can see it is having a steroidal background and as well as it is having a triterpenoid background in their aglycan part. So secondary metabolites obtained by cholesterol metabolism, just as a schematic diagram it is shown in this slide, that is Squalene can give rise to pentacyclic triterpenoid beta amyrin molecule, and also cholesterol can give rise to trigogenin, diosgenin, which is very common sapogenin, which we will study, dioscoria, which contain diosgenin, which is also having more action towards even anti-cancer effect is also there, and it has many uses are there. And also you can see emogenin and sapogenin. These all are some of the example, very few I have denoted. And then when you see 2022 dihydroxy cholesterol, that means 20th position, 22 position of the cholesterol, dihydroxy cholesterol containing 20th and 22 position. You have to know about cholesterol structure, please. I have not mentioned here, but you have to know. And that place, and even numbering also very important for the student should know, because in the future it will be helpful for you in other chemistry subjects also, and even in the synthetic chemistry or in the medicinal chemistry it will be more useful for you to study. And you see this 2022 dihydroxy cholesterol is now converted into pregnenolone. So pregnenolone already structure is shown in that, even cholesterol also shown in your previous classes. You can just refer that. And when the addition of 3 carbon atom units, you will get these compounds which is listed out. I have made a oval shape. There is no space. That is why I put an arrow this side, and it is shown here. That is digitoxygenin, jitoxygenin, jitoxygenin, and strophanthinidin, which is strophanthus, which is another cardiac glycoside. Strophanthine seeds will contain this strophanthinidin. So this is the thing. At the same time, when you are adding 3 molecules of carbon atom units, and it gives silaranin. Squill is having this one, silaranin. It is also having many, it is used in rodenticide, and it has many other cardiac activity also. It's a cardiac, it comes under cardiac glycosides. And siliglacosidin and helibriginin, these all are some of them which is formed as a secondary metabolite from squalene. So squalene, how it is formed? It is also passed through it comes from the acetate pathway. So basic acetate pathway, and then involved many shikimic acid pathway to give many structures of secondary metabolites, and you will form these glycosides. So student have to explain clearly if you understood what is the basic metabolic pathway, how they are involved in production of various secondary metabolites, especially these glycosides. And see here, the aglycone or isothiocyanate glycoside. There is another glycoside, isothiocyanate. That means NCS, isothiocyanate, cyanate, isothiocyanate glycoside may consist of either aliphatic derivative biosynthesized via acetate pathway or aromatic derivative produced by biosynthesized by shikimic acid pathway. So aromaticity can be done only by shikimic acid pathway. So it can undergo aliphatic, there is aliphatic derivative, and also it can go for aromatic derivative using shikimic acid pathway. So acetate pathway can give rise to CH2 double bond CH, N double bond C, and with one sulfur with glucose, and OSO3K. And phenylalanine can also convert into N-hydroxy phenylalanine, and it can be converted into phenyl acetaldehyde oxime, which can give rise to glucopropylene. And glucopropylene is gas C2H5, CH2, N double bond CS, and you are getting that isothiocyanate molecule, which can give rise to even combined with a glucose molecule in a combination of a linkage, and so that you will get a glycoside. Isothiocyanate glycosides can also be synthesized. And here this is a chart and also a schematic diagram which is showing that general scheme of amino acid formation. General scheme of amino acid formation. Glucose. From glucose, we know that glucose and erythrose 4-phosphate will give 7 heptilo compound, and it can give rise to even next step. It is possible before going to the heptilo, it can give rise to histidine, Tyrosine phenylalanine can be formed by means of shikimic acid pathway. These all compound which we have studied. And then pyruvate can give rise to alpha ketoglutarate and glutamate, and it can give rise to proline, glutamine, ornithine, and citrulline, and arginine can also be produced. And from alpha ketoglutarate you can produce even homoserine, methionine, and threonine, and isoleucine, and also directly you can get aspartate and aspargine can also be produced. And from this even a product of diamonomalate, pimelate, and it can give rise to lysine also. So you see pyruvate can directly give rise to valine, leucine, serine, cysteine, alanine, and tryptophan. These all the aliphatic and aromatic amino acid generally in the schematic diagram will give you some idea to remember how they are formed and how they can be produced. And this is one structure. Some of them they used to ask how cysteine and glycine and cysteine or serine and glycine and cysteine are produced by biosynthetic pathway. You can see 3-phosphoglyceric acid is converted into 3-phosphohydroxypyruvic acid, and this is converted into 3-phosphoserine, and then it is converted into serine, and that serine can be converted into glycine, and serine can also convert it into alpha acetyl serine and be to the presence of thiolase, thiolase it is, and you will get cysteine because sulfur containing amino acid is cysteine will be formed. And this is another one you have to remember, biosynthesis of amino acid. Altogether ornithine is a plant amino acid. You might not have come across this ornithine when you are studying biochemistry. This is purely plant. Ornithine is an amino acid. Proline, hydroxyproline, and arginine. These all things can be arginine, G is there in arginine. So this arginine also can be formed. So you see that is alpha ketogluteric acid converted into glutamic acid. Glutamic acid can be converted into glutamine. And here glutamic acid can be converted into glutamic semialdehyde, and it can be converted into ornithine, and then it can be converted into arginine. And proline 5-carboxylic acid converted into proline and hydroxyproline. These all are the some of the products which can be obtained from this area. And that is the small class which we were trying to finish today, but we had lot of disturbance in the internet, so that we were unable to execute in the live class. So kindly students are requested to download this and listen to it, and also study by writing. The structures you have to write and study, otherwise it will be difficult. And also now I would like to tell few words which I don't know how far all students have listened to that my advice. Please try to utilize this online class which is arranged by our institution, and also our teachers who are taking more effort to prepare all their online classes with great difficulty. So kindly students, always whenever the teachers are waiting and having some problem with the internet and they are finding difficulty, you please be patient and don't chat yourself, don't unmute your audio, and don't do that please. And also try to inform the teachers and try to be supportive to them. And also when they want to ask you any doubts, if they want to clear, that time people there are many students who are unable to even to leave the classroom because they are just putting on their instrument and they are going outside. So don't do that. When you are in online, please cooperate with your teachers and your colleagues, and also make our online classes successful. Thank you very much. Thank you very much for attending this class, and next class we will see the second unit. Thank you.